Hey, welcome everybody to a new global IMMUNOTALK. I'm Carla Rothling and I'm here with my co-organizer, Elina Zuniga. It's a pleasure to welcome you. Just as a brief reminder, we have organized this to bring outstanding talks in immunology in an egalitarian manner to all of you across the world and without the need of traveling. Of course, we couldn't do this without our extraordinary speakers. We will hear today from Laura Mackay, and Elina will tell us a little bit more about that. And we look forward to having Ido Amit from Israel next week. So with this, I will let Elina introduce our global immuno speaker for today.
Thank you, Carly. As you know, we have a very different time zone with Australia, so we pre-recorded this session. So I will share now the screen with the recording so everybody can enjoy the Laura's talk. Okay?
Indeed, it's 4 a.m. in Australia.
Okay, it should go now.
Okay, hello everyone. I'm pleased to join this pre-recorded session with Dr. Laura Mackay. Okay, although she now resides in Australia, where she is professor at the University of Melbourne. And she completed her PhD at the University of Birmingham in UK in 2009, followed by a postdoctoral training in the laboratory of Professor Francis Carbone in Melbourne, Australia. She started her own group in 2016, just 4 years ago, at the Peter Doherty Institute in Melbourne, where her laboratory studies memory T-cell responses, infection and cancer. Laura is currently Howard Hughes Medical Institute and Bill and Melinda Gates International Scholar, a Sylvia and Charles Virtual Charitable Foundation Senior Medical Research Fellow, an NHMRC Leadership Investigator, as well as a Dame Kate Campbell Fellow. Among the many contributions that Laura has made to science, I would like to highlight today her fabulous work on the characterization of tissue-resident memory T-cells, also known as TRMs, which reside in different tissues such as the gut, skin, lung, and constitute one of the first line of defense upon secondary infections. In particular, Laura's work greatly helped understanding the developmental precursors of TRM cells, the transcription factors such as TBET and cytokines such as TGF-beta and interleukin-15 regulating TRM cells, as well as the role of these cells in different settings and their capacity to locally proliferate upon reinfection. Her research not only significantly increased our basic understanding of this unique and important population of memory cells, but also bring us closer to ultimately manipulate them to better fight infections and tumors, which I understand is a major goal of Laura's laboratory. So not surprisingly, given her fantastic work, Laura has been recognized by awards, including the Prime Minister's Prize for Life Scientist of the Year, the Gottschalk Medal Award by the Australian Academy of Sciences, the Eureka Prize for Outstanding Early Career Researcher, the Woodward Medal in Science and Technology, and the Michelson Prize for Human Immunology. And I should probably stop there so we have enough time for the talk. So apart from these amazing accomplishments, Laura finds time to hold leadership positions in science, and we're very thankful for that. And she is currently president of the Federation of Immunological Societies of Asia and Oceania. So Laura, I cannot emphasize strongly enough what an honor and pleasure is to host you as a global immuno speaker today. We really appreciate your time to prepare this talk, and we look forward to it.
Thank you so much for that incredibly kind introduction. And it's an absolute pleasure to be a part of Global IMMUNOTALK. I absolutely love this initiative. and the idea of bringing free science to people all around the world, people who perhaps couldn't get to see and get the exposure to immunology. It's absolutely fantastic. So thank you so much.
Thank you, Laura. We're happy to do it, and we can only do it because of the generosity of the speakers like you. Thank you so much. So before we move to the talk, we would like to ask you a question. And today we have a question for you that is, what is the advice you would give to your 25-year-old self? So if you could go back time, what would be that advice?
When I was 25, I was just starting my postdoc, and being a postdoc, it's an incredibly challenging time. I think I would try and tell myself to enjoy it a bit more, because it's really when you're on the front line of making the discoveries, and it's sort of just, you know, a really unique opportunity in life where you can really follow your passion, and just, it's so much fun. Like, before you get sort of bogged down in some more of the bureaucracy, you can just focus on the science. And part of enjoying it a little bit more, also, I tell myself to speak up quite a bit more. Sometimes you think that, you know, you're not the smartest person in the room and you shouldn't ask the stupid questions. Whereas now I think, you know, I'd say part of enjoying it is that you're there to learn, everybody's learning. And so that's what I would say.
That's beautiful. Thank you so much, Laura. Yeah, enjoy the journey, right? Sometimes we are too focused on the goal and we forget to do that. Thank you, I appreciate it. So if you like, you know, you can share your screen.
Absolutely.
Fantastic. Perfect. Now it works.
All good?
Yes.
Fantastic. So you gave TRMs an excellent introduction. Thank you for that. And today I'd like to talk about some of our new insights into TRM biology. I'd first like to talk a little bit about why we work on these cells and some of their protective functions, and then give a couple of new stories we have talking about the influence of the tissue on instructing TRM cell fate. So just to start off with just an introductory slide just for our junior immunologists in the audience. We are interested in our lab on the generation of memory T cells. And actually, I might just get my laser pointer up here. Actually, can we see my arrow?
We can see your pointer as an
Perfect.
arrow. Yes.
Thank you so much for that. So when you, for example, if you get an infection with a virus, you'll get an expansion of the affected T cell pool when a naive T cell recognizes antigen. And then, you know, after days to weeks following the infection, after the resolution, that T cell pool will contract. It'll undergo more than 90% of contraction, and then you'll be left with this long-lived memory T cell pool. And these memory T cells can provide re-encounter protection if you get a re-encounter with that same pathogen. And inducing memory T cells is one of the primary goals of vaccination. So you have memory T cells in the body specific to a given antigen or pathogen, which can effectively provide immunity. And what we now know is that not all memory T cells are equal, and the memory T cell pool has a lot of heterogeneity within it. So if you look in this memory T cell pool, you'll have three flavors as defined at the moment, called effector and central memory T cells, as defined several decades ago now. These cells will be circulating in the blood, and also defined about 10 years ago now, our tissue-resident memory T cell pool, or TRMs. And these cells permanently reside within tissues of the body and characteristically are non-recirculating, so they can't be found in the blood. So we can really segregate our memory T cell flavors by the properties of migration and the organs in which they can be found. So our effector and central memory T cell pool, if you take a blood sample, for example, that's what you'll find. You'll find long-lived effector cells specific for a given antigen, but also our effector memory T cells. And a lot of the sort of initial studies on memories, particularly in humans, failed to encapsulate sort of, I guess, the diversity of T cell memory by not looking in peripheral tissues, simply because it's pretty hard to get access to, say, a piece of healthy human liver as opposed to just getting a blood sample. But if you do go, apologies for that, inside these organs, you can see a different flavor of T cell memory, and that's our resident T cells that are embedded within organs such as the skin epithelia and also the epithelia of the intestine. And if you look in solid organs like the liver, which are in contact with the blood, which is always recirculating through these organs, you can see you would have 3 flavors of memory T cells within an organ like this. The population that doesn't reside are TRMs, and also our central and effector memory T cells. So TRMs, they're a non-recirculating cell type. They're made permanently in barrier tissues. They've predominantly been characterized of the CD8 positive T cell subset, but also CD4 T cells can establish residency as well as lots of other immune cells like NK T cells can also establish permanent residence within a cell. These cells have now been found in pretty much all organs in both humans and mice, and they are found in frontline organs such as the skin and the gut, as I mentioned, but also populations of non-recirculating cells can even be found within the spleen and the lymph node. And these cells are distinct from circulating memory T cells in their phenotype, morphology. I've just got a little picture here of those cells in the skin. They almost look like Langerhans cells, squashed by their location, and their developmental pathway, which we've worked on for many years now, is also distinct from circulating T cells. So how to define these memory T cells? Really the gold standard to show that a TRM is truly permanently embedded within the skin is either tissue transplantation, or the gold standard in mice is parabiosis. So 2 of the first studies by Tom Gebhard and Frank Carboni and Dave Mazepust and Lefrancois defined that if you took either the skin or a piece of the intestine from one mouse that had embedded memory populations within that tissue and you transplanted that onto another mouse, you could find TRM from donor origin within that transplanted tissue. So within transplanted tissue, you'll find these donor TRMs, as I've shown in this cartoon here, and then you'll get new recruited TRMs from the new recipient that will come in and convert to the TRM phenotype if they've been stimulated. And this has more recently been shown to be true in humans. It's been shown by Donna Farber and others that you can transplant tissues and find cells of donor origin within those grafts for several years following transplantation. And here, this is, as I mentioned, the gold standard for a mouse biologist. If you've got one parabiont that's been previously infected, say in this case, it might have been a respiratory infection, so you'd have TRMs lodged in the lung. If you can join the circulation with another recipient mouse that would have a different congenic label, the circulation will go into equilibrium between these 2 parabionts, but you will see the resident memory T cells sticking around within just this donor here. And so the way, if you can't do these fancy tricks, that you would define a cell to be resident is by their cell surface phenotype. And there's pros and cons to this. CD69 will encompass the majority of resident memory T cells, but it's not a sort of be-all and end-all marker. We generally say for TRMs in combination, just because CD69 will also label recently activated cells or cells chronically stimulated within a tumor microenvironment. But certainly the majority of TRMs do express this marker, and they can be found within all tissues of humans and within mice. And transcriptional signatures have also been used to define resident memory T cells. And a lot of genes can be found within common to this cell population within all organs, which I'll touch on a little bit later. So what I like to sort of raise pretty early on is, why should we care about this distinct population that's within tissues? And that's because it's been shown by many studies now over the past 5 to 10 years, is that these cells are critically important in protecting against a variety of infections. They've also been shown to be able to control cancer, and are associated with a better prognosis and outcomes against certain cancers. And also because these T cells are embedded within tissues and in the front line, it's perhaps not surprisingly that they're associated with various autoimmune conditions within tissues as well. And so targeting these T cells specifically within tissues is of great interest for generating new immunotherapies and vaccines. So just to give you one such example on how TRMs can protect against infection, and this has been shown by a multitude of groups. If you have TRMs in one tissue, they can protect you against reinfection with the same pathogen. But in this example I'm showing you here, if you take a completely naive animal and you embed T cells within either the peripheral tissue of that animal, in this case, the skin, or if you generate populations of circulating memory T cells, which can be found throughout all tissues. And so you have a setup here where you have TRMs embedded, in this case in the skin, or you don't. They have to be specific for the pathogen of interest. And then, sorry, I keep doing this. And then you challenge with that pathogen. In this case, HSV, which is a skin infection. You can see if you've got TRM embedded within the skin, right place, right time, that's where you're challenging with pathogen. If you've got the TRM, you have the benefit of being able to immediately block the infection at the front line. Whereas even if you just boost huge numbers of circulating T cells and then challenge those mice, those circulating T cells have to infiltrate the tissue, and that delay results in sort of an inability to protect against this skin lesion. So the amount of protection you can get from TRM cells is directly linked to the number that you can lodge within the tissue. And here we're just showing a graph here. This is work by Simone Park, published a couple of years ago now. The more TRMs that you can get in the tissue, if you then challenge those mice, you can see a decreasing viral load against HSV as measured by PFU. And in humans, there's evidence as well. This is a nice example from Larry Corey's group, that if you have high numbers of TRMs within the genital mucosa, that correlates with subclinical, that means non-presenting HSV reactivation. So it's been shown over the past, let's say, 2 to 3 years now, that if you are able to embed TRMs into tissues using various strategies, they're able to prevent cancer growth following challenge with a range of tumors, including B16 melanoma, MC38 colon cancer, and also head and neck cancer. And these TRMs can also protect with the in the absence of the circulating population. So here's just an example also from Simone Park in a study led by Thomas Gebhardt at the University of Melbourne. And you can see here, if you can generate populations of TRM, or both TRM and circulating T cells, these would be, these T cell populations would be able to be recognized by the tumor, so they would be specific. And using various tricks, which immunologists have worked out how to do now with TRM, if you just have TRM within the skin of the mice, you challenge with B16 melanoma, you're able to protect, the mice can remain tumor-free. And that same protection is afforded when you both have resident and circulating population, showing that you can have afforded protection just by the resident population by itself. And here is compared to naive mice that lack specific T cells. And as I mentioned, these populations, at least with the TRM cell phenotype within humans, have now been associated with better prognosis against various cancers, including ovarian, lung, and breast cancer. And here's just another example here showing that TRMs can protect, can better predict survival than a total CD8 positive TIL in triple negative breast cancer, publishing this together with Shireen Loy a couple of years ago now. And showing that if you generate a signature that's associated with resident memory T cells, it's better associated with disease-free survival. So these patients that have a high TRM score, as opposed to those that are lacking cells of this phenotype, as compared to total TILs, which have been long associated with a better prognosis against triple negative breast cancer. And so, and just lastly, one of the other functions of TRMs that I mentioned is as they've at least been correlated with various autoimmune pathologies, particularly in the skin, where you might think that makes sense, because a lot of skin pathologies often reoccur at the same place at the same time, where you would have these TRMs fixed within location. And so conditions such as vitiligo, where you have melanocyte destruction and loss of pigmentation, and also psoriasis, where you have keratinocyte hyperproliferation, have both been associated with high numbers of TRM cells that can produce either interferon gamma or IL-17. And also various other disorders have been associated with TRM, such as colitis, diabetes, and MS. And recently we showed that even if you just have a fixed sort of lesion within the skin, and this is from intradermal drug testing, you can have an embedded population of skin TRM that persists within a previously inflamed site. So here's just an example here. This drug testing was performed by clinician Jason Trebbiano, who was testing patients for reactivity to certain antibiotics. And you can see here there was a reaction in this lesional site, no reaction in this control here, and then coming back several months later and analyzing the T cell populations within these sites, and you can see a maintenance of resident memory phenotype cells within the skin of a previously inflamed fixed skin site within human patients. So with regards to how TRMs can protect, a lot of work has been done by our group and others showing across the years how these TRM cells can protect or cause pathology in a wide range of settings. And certainly one of the major functions of TRMs, potentially rather than direct killing of tumor-infected cells, even though there's some evidence they can do that, but it's largely thought that their major function is releasing bucket loads of cytokines. So interferon gamma, IL-2 and TNF, also IL-17 in pathological settings, and some seminal work by the groups of Mazepus, Schumacher, and Akiko Wakasaki showed that this could create an antiviral state within the tissue. Then that would result in the recruitment of many other immune cells to the front line, such as B cells, dendritic cells, and NK cells. And so that would bring in a lot of other cells into the tissue where the TRMs are embedded, resulting in superior protection. Also, what happens when a TRM cell can get restimulated, they can locally proliferate. There's evidence they can survive in situ after the challenge, give rise to new TRM populations. And of course, following a secondary encounter, new resident memory T cells will be seeded alongside those pre-existing populations by secondary effector T cells that will be stimulated and coming into that site. So for the remainder of the talk, I'd really like to talk about how these T cells are generated, given that they're so important in a variety of conditions. And so just to give you an example of the types of memory T cells and what they look like following a viral infection. So the skin infection with HSV is one of the primary models that we like to use in the lab. And we use the skin infection model in combination with transgenic T cells that are CD8 positive and specific for the immunodominant epitope within herpes simplex virus. And generally they'll be tagged with a congenic label such as CD45.1, so we can track the priming and differentiation of these T cells within a new host. So if you look more than a month following primary infection, and you look in the spleen, you look in the infected site, which is the skin, you can see a persistence of the memory T cells, our GBT1s. They express CD45.1. This is their TCR receptor, the alpha-2. And you can see if you go within the spleen, you can see a diversity of populations, our long-lived effector cells, and also our central and effector memory T cells. And you can see that these molecules lack expression of the molecules CD69 and CD103. In contrast to those GBT1 cells we can find embedded within the skin, which are double positive for both these markers. And CD103 is often utilized as a canonical marker of TRM, at least within epithelial tissues, along with CD69, which is shown to be present on the majority of this population. And getting back to just thinking for a second about those fixed eruptions or lesions that you might see in the skin and the fixed location of those TRMs that stick around, here's just an example tracking the CD8 positive TRMs that we see in mice following the resolution of infection. So if you look within a previously infected HSV site, you will see that these CD8 TRMs, they'll come in, they're denoted here in red within the lesion. And of course, there'll be many other populations within the skin, and you can see gamma deltas here in green, which will be on either side. So it looks like the TRMs are coming in and displacing other populations as they influx into the skin. But importantly, and this is the point that I wanted to make here, is that if you look 30 days after infection or almost a year after infection, and you look for the density of T cells within the skin, they really remain remarkably constrained to that previously infected site. So here we're just moving out just a few millimeters either side of the lesion, and you can see that those T cells remain fixed. And here's just a sort of 8-hour movie of the movement of resident memory T cells, in this case shown here in green. And you can see that these T cells are always patrolling, but again, they're very fixed in location. And you don't really, if you track their migration patterns, they remain remarkably localized to the site in which they're embedded. So another model that we'll use and we'll use throughout the talk is a systemic infection, acute infection with LCMV, which is most favorite infection model of a lot of immunologists out there. And we'll use this again in combination with transgenic CD8 T cells that are specific for this pathogen. And if you go and you look throughout the organs at the expanded P14 population more than a month following infection, and you can see here are our transgenic T cells here, and you look at the pattern of CD69 and CD103 within different organs, such as here we're looking at the small intestine within the intraepithelial layer, the liver, the salivary gland, and the kidney. You can see there's a spectrum of expression of CD69 and CD103. With those, just like the skin, the epithelial tissue here is double positive for these markers, whereas solid organs such as the liver and kidney exclusively express CD69 and lack populations of CD103. And the CD69 population within organs such as the liver has been shown to be truly resident by methods such as parabiosis. And one point I wanted to make was the skin, you don't just, in general, to get a decent number of cells within the skin, you have to use an inflammatory stimulus. So if you just throw effector T cells to a mouse, in this case, this mouse is not infected, so you just put effector T cells into the mouse. Again, they can form populations of resident memory T cells, which have the same phenotype as those after infection. So here, there's that double population of CD69, CD103. Again, cells in the liver expressing CD69. But for the skin, to get a sort of a decent amount of population to enumerate the phenotype, you have to add a inflammatory agent such as DNFB, which we commonly use in the lab to pull those cells into the skin. And you can see that they upregulate that phenotype CD69, CD103 high, again showing that that phenotype is linked to the microenvironment and not necessarily recognizing a local antigen within the tissue. So a lot of the phenotypes that we see are really dictated by the tissue and not by recognition of antigen or anything to do with the infection. So certainly, even though I've shown you that there are differences between, at least for the expression of CD103, that's distinct between organs, a lot of TRM cells, regardless of the tissue and regardless of how they were generated, infection or not infection, they have a shared genetic signature. And several years ago now, we identified a signature which was common to TRMs that we isolated from different organs, such as the skin, lung, and gut, and they were induced by different pathogens. In this case, we used HSV, influenza to generate TRMs in the lung, and LCMV to generate those in the gut. And you can see here, if you look at the transcriptomes and you look on this PCA, you get a clustering of the resident population and a clustering of those T cells that circulate and can be isolated from the spleen. So what you can see is that our memory T cells in the spleen actually have more in common with naive T cells that haven't been primed by antigen than those within the tissue. And this core signature is common not just to CD8 T cells that prime for infection, but a few years later, we extended this looking at natural resident populations, if you like, such as resident NK T cells in the liver and also tissue resident NK cells, as well as CD8 populations. In this case, we also included a non-epithelial site such as the liver. And you can see again a shared signature between these resident populations. And a lot of what's in common to these populations is the upregulation or downregulation of molecules associated with tissue egress. So you can see here in our downregulated genes, downregulation of genes such as S1PR1, KLF2, and S1PR5. And the signatures that we find in mice can also be enriched within signatures found within human TRM. And recent work by Donna Farber has shown that if you take, I'm sorry that keeps happening. If you take a human TRM signature and you look and you compare 69 positive cells from both human and mice, you can see them clustering together here within this cluster here as compared to those cells that are circulating from both mice and humans. So I'm just thinking a little bit about TRM development. And as I mentioned, this is something we've worked on for a long time now, and there's certainly multiple steps for a T cell to go into a tissue and differentiate into a resident memory T cell. But one comment that I wanted to make here is that shutdown of tissue egress is certainly one process that's common. Again, the downregulation of KLF2 and S1PL1. Also, to get into the tissue in the first place, you need to upregulate certain chemokines such as CXCR3, CXCR6, and then a T cell needs to make the decision of whether it's going to stay, whether it's going to go within a tissue. Certain transcriptional regulators such as Hobbit and BLIMP1 are important here in suppressing the pattern of tissue egress. So these transcription factors have been shown to drive TRM formation across multiple tissues. But of course, there is a lot of heterogeneity aside from this core signature, which can be instructed by the distinct microenvironment in which a TRM is embedded. So once a T cell gets into the epithelia, then there's a second wave of the TRM program, which will really cement that T cell's fate. And over the years, we've shown that certain cytokines and transcription factors are important to these processes. Some are true of T cells within many tissues, some are only true of certain populations. And so I'd like to start to articulate some of the heterogeneity we've been exploring with you now. So we've been doing some high dimensional profiling of resident T cells in different tissues, and this is looking by flow cytometry at what's expressed on the surface. And this work's been led by Max Everard in the lab. So what Max does is he will generate populations of resident T cells or memory T cells throughout multiple different tissues, and they'll be induced by LCMV. He'll then barcode these cells using different strep-avidin combinations, throw them all in the same tube, and stain all these P14 T cells with a backbone panel using certain markers that are associated with different T cell differentiation states. And then Max has employed a legend screen, which has multiple unique surface molecules all labeled on the same fluorochrome. And then he uses an infinity flow analysis pipeline to see how the expression of multiple markers within a given T cell population. Now, if you look by UMAP at all our P14s here, as defined by our backbone panel here, you can see you'll get a cluster of TRM cells, and this is regardless from what tissue they're coming from. So the liver, the spleen, the gut, for example. You'll also see a cluster of our circulating T cells. These are effector and central memory T cells, as well as naive T cells. And what you can find if you look at predicted expression profiles is here, you'll see a little of what you expect. You'll see that our TRM cells from different tissues are clustering on markers such as CD69 and also CXCR6. And you can also see the segregation here of populations that express CD103 and populations that do not. And here, this would be the gut population and our sort of mixing of our 103 positive and negative populations within the salivary gland. And what you can see here, if you just look at expression profiles of CD69 and CXCR6, higher expression on TRMs globally, regardless of tissue, compared to our circulating and naive populations. And what Max has been doing is starting to explore the heterogeneity that comes down to TRMs that come out of this cluster within a different given tissue here, such as our CD103 expressing gut population here, and looking at different molecules that are associating correlations of these populations, identifying unique markers that can be used to identify TRM cells within unique organs. And one step further, you can go into the circulating population now and show that these cells can also be broken down by into several different clusters, which can be identified by unique surface molecules, and also also going into a distinct tissue such as the liver, which will harbor, as I mentioned, multiple populations, such as resident memory T cells, which can be defined by CD69 and CXCR6, and also our long-lived effector T cells, defined by CX3CL1, for example, and memory T cells. And this is something that Max is exploring at the moment. So what I wanted to do was try and give a couple of examples of how such heterogeneity within the TRM population can affect their behavior and also affect their function. So I want to present a couple of stories now. And the first was published a few months ago by Hannah Frizzle and Ayesa Concesa in the lab, looking at the spectrum of fatty acid binding proteins expressed by different TRM populations within distinct tissues. So it's known that memory T cells will utilize endogenously synthesized fatty acids for their survival and for energy. And a really nice paper came out from Tom Cooper's lab a couple of years ago now, showing that TRM survival and function specifically requires endogenous fatty acid uptake, and also that TRMs formed within the skin are dependent on Fab4 and 5. Now, given that there's a spectrum of fatty acid binding isoforms that are known, we wanted to ask the question of whether all TRMs sort of within different tissues would have similar dependencies and express similar expression profiles of fatty acid binding proteins. So what we did was we infected mice with various pathogens, including HSV or LCMV, and we looked for the 9 different isoforms of Fabs from 1 through 9. And you can see, akin to what the Cooper laboratory found, we see in skin TRM, in this case generated by HSV, but also by vaccinia. Also, if you throw effector T cells in, bring them in by DNFB, T cells within the skin will express Fab4 and Fab5. But if you look in different tissues now, such as the liver and the small intestine, you can see a different spectrum of Fab expression. Those in the liver predominantly express Fab1, and those within the gut express multiple Fabs, identifying with a profile of Fab1, 2, and 6. And what we found is that this expression of Fab sort of profiles was true not only of CD8 T cells following LCMV, just shown as a heat map here, but also if we look at endogenous immune cells in tissues, such as resident ILCs, NK cells, dendritic cells, NKT cells, and gamma deltas. You can see those populations within the skin here, looking at ILCs, Langerhans cells, and DETZs, predominantly express Fab4, just like the CD8 T cells. Also similar expression profiles for those isolated from the gut and the liver, which predominantly express Fab1. And so what this really shows is it's really the tissue microenvironment that's important rather than the cell type when it comes to expression of different Fabs. And so we went further to show that it was actually the tissue that could induce Fab expression, here by just taking in vitro cultured CD8 transgenic cells and effector cells. After you've bolstered them in culture, they will have negligible expression of Fabs. And so what we did was we took these T cells, we took various organs such as the spleen, liver, and skin, homogenized these organs, took the supernatant off, and just threw this onto our T cells and looked a few hours later. And what we found is that these effector T cells would upregulate a fatty acid binding profile according to the tissue supernatant which had been thrown onto these T cells. So taking supernatant from the liver, you can see expression of Fab1, but not other Fabs, and we look for the whole range of Fabs, but I'm just showing you here, so you can see no Fab2, 4, or 5. 4 is always very negligible for the liver. Whereas if you take supernatant from the skin here, you can see high expression of Fab4, specifically when you throw on skin supernatant, but not Fab1, showing that it's really something within the tissue which is driving the upregulation of this Fab expression. Now, it's recently been shown within, I guess, the past few months within the field that TRMs are more dynamic than we originally thought. And they can actually start to re-express certain molecules if they're relocated, and they can be re-stimulated. And so we wanted to ask if we isolated resident memory T cells, in this case from the liver, put these into new hosts. So here we're isolating P14s that are CD69 positive from the liver, putting them into new hosts, which we then infected with LCMV. And then we looked what these ex-liver TRMs could give rise to. And we looked for the change in their fatty acid binding protein profile. So we isolated P14s that were originally ex-liver TRM, and we could identify them from the congenic label that they were expressing as they went into new hosts and expanded. And we looked within the liver, small intestine, skin, and spleen. Firstly, just showing that these cells can re-expand when you put them into new hosts and challenge with LCMV. And here, just reminding you the pattern of Fab expression associated with various organs. So these ex-liver TRMs would have originally expressed Fab1, now they've been re-stimulated, and we're isolating them from these various organs. And what we can find is that those ex-liver TRMs, we're looking either 7 or 30 days following infection. If they've gone to the gut, here they're expressing Fab1, 2, and 6, the expected profile. And if those ex-liver TRMs are relocating into the skin, in this case, we use DNFB to drag them in there, they are upregulating Fab4, showing the dynamic expression of Fabs upon relocation of a TRM to different tissues. And also, not showing the data here, but they also became dependent on that Fab. So whereas we know that skin TRMs are dependent on expression of Fab4 and 5, we showed that liver TRM are indeed dependent on Fab1. Now, the last experiment I showed you was following re-stimulation after infection. And so we asked, was that necessary? What if you just took isolated resident memory T cells and transferred them directly into the tissue with no re-stimulation at all? And so in this case, we used a different setup where we transferred effector OT1s into a mouse, allowed for resident memory T cells to be generated, isolated those CD69 positive TRMs from the liver, and transferred them epicutaneously directly into the skin. And so these liver TRMs that would be originally expressing Fab1, we asked what would they look like if we put them just directly into the skin, and we looked a few days later. And what we can find is that these ex-liver TRMs in skin indeed switch from expressing Fab1. These are our original liver TRMs, and Fab expression in the liver is, as you can see, much lower than the screaming high Fab expression that you get in the skin. And you can see here a regulation of Fab4 and Fab5, showing that these ex-liver TRMs downregulate Fab1 and upregulate 4 and 5 upon relocation to the skin. So just the last story I wanted to show just shows you some more of the implications of responding to different cytokines and being in a different tissue on certain behavioral properties of resident T cells. And this work is a recent story, but it's been quite a long and really heroic effort by postdoc Susan Christo in the lab. So as I've shown you, TRMs within different tissues have different profiles of at least CD103, for example. And if you throw TRMs boosted by infection, generated by infection from the skin, saliva gland, and liver, you can segregate them based on CD103 expression. So our skin population exclusively CD103, our salivary gland has a mix, and our liver cells lack expression of this molecule. And really, when we think of the spectrum of TRMs, and generally in the lab, we now look at TRMs pretty much in all tissues, which is very laborious for everybody involved in that work. But the skin and the liver are really the polar opposite extremes of TRM phenotype that we see. And so just looking at a variety of molecules that are differentially expressed between liver and skin TRMs, such as CD103 as an example here, I'm just showing the spleen as a comparison. If you look at certain adhesion molecules, such as E-coherin, it's up with the skin, TCF1 downregulated on skin TRM compared to those in the liver. And also, we see upregulation of certain immune checkpoints, specifically on skin TRM, as compared to those in the liver. And this spectrum of molecule expression, again, is not to do with how these T cells were generated, because we see exactly the same profiles in the absence of infection. So this maintained PD-1 expression on skin TRM is not because these T cells are constantly seeing antigen, because if we just look at T cells that are embedded within naive mice, take in vitro activated OT1s, throw them into mice, bring them into the skin, you can see exactly the same expression profile. So this heterogeneity doesn't just extend to phenotype, but something I was alluding to is actually extends to the behavior and the functional capacity of these cells. So if we isolate both skin and liver TRM out of mice, and in this sort of experiment, we were trying to get at this for several years, but we really had to come up with a scenario whereby these cells are isolated in exactly the same way. So we would use a non-standard skin isolating profile, for example. We take equal numbers of skin and liver TRM and stimulate them with peptides. In this case, I'm using OVA because we're using transgenic OT1s, and just look at the amount of cytokines and the amount of polyfunctionality that these TRMs can have. you can see a far superior polyfunctional capacity of liver as compared to skin TRM. And also, if you just look at the half-life of skin versus liver TRM, again, you can see stark differences. So we're looking within the same mouse at the same type of T cell that's been embedded with either the skin or the liver. And you can see that skin TRM populations are maintained within the tissue for long periods of time, whereas liver TRM, even though they are resident, and they were done parabiosis to confirm this, over time these cells will decay, having a far shorter half-life than those within the skin. So I'm showing there's a reduced functionality of skin versus liver TRM, but there's also an enhanced longevity of skin versus liver TRM cells. So what we know is we've been working on TGF-beta for a long time in the lab, particularly in the context of skin TRM. We know it's absolutely critical for skin TRM formation and also their maintenance within tissues. But something that we found, I guess, a few years ago now is that TGF-beta knockout cells can survive within the liver just fine. So again, here in this sort of setup of an experiment, we're transferring cells that are wild type or deficient in their capacity to respond through TGF-beta. And then looking forward 14 days later, you can see a decay of the skin population as we'd expect, but a maintenance of cells within the liver. And here just showing a ratio fold change. We're losing cells in the liver, in the skin, but those in the liver and spleen are just fine. But TGF-beta isn't just required for the long-term survival of our TRM population or its formation, but it's also important for driving a lot of molecules associated with the identity of skin TRM. So not just CD103, which is well known, but others such as PD1. So you can see here that whereas if you look between wild type and knockout cells within the skin, you can see that if you look at our knockout cells here that can't respond through TGF-beta, TGF-beta is driving this expression of CD103. But if you look in the liver where this molecule is barely expressed, there's no change whether you're signaling through TGF-beta or not. And again, you can see no change in the phenotype of cells within the liver. And so it's not just their survival, it's their phenotype is unaffected by TGF-beta signaling. And we know that TGF-beta can drive a lot of transcriptional programming associated with the skin. It's well associated with CD103, but it drives an absolute slurry of molecules specifically expressed within skin TRM. And TGF-beta alone pretty much accounts for a lot of the differences that we see between skin and liver TRM. So in this sort of in these gene set enrichment analyses that I'm showing you here, we take our RNA sequencing data from wild type or TGF-beta knockout cells isolated from the skin, and we look at the top genes that are either up or down regulated in our wild type or knockout cells. And then we compare those gene sets to establish TRM published signatures from skin or liver TRM. And what we find is that if you look at those genes associated with skin TRM, they're up in the wild type, and those that are down in skin TRM are down in wild type cells that can respond through TGF-beta. But importantly here, if you look at genes that are upregulated in liver TRM, they are up in cells that cannot respond through TGF-beta, and also molecules downregulated in liver TRM are down in TGF-beta knockouts, showing that our liver TRM transcriptionally resemble those skin TRM that cannot respond through TGF-beta signaling. So they're transcriptionally similar to skin TGF-beta receptor 2 knockout cells. Now, TGF-beta, yes, it regulates a lot of the phenotype, transcriptional identity, but it also seems to act as a handbrake, restraining TRM function specifically within the skin, whilst having no effect within the liver. And so in this sort of experiment, again, we isolate wild type or TGF-beta deficient cells from mice. We take these cells out and we throw them in culture with specific peptide. And what you can see now is that if you look at our wild type and cells that can't respond to TGF-beta signaling, you can see that TGF-beta is really acting as a handbrake here, and it's dampening our skin cell function. So you can see that, as I showed you before, we have a poorer capacity of skin versus liver TRM to produce cytokines. A lot of this is dictated by the capacity to respond through TGF-beta. So lastly, I just thought I'd show you that these liver TRMs, they can adopt the phenotype and dependence of skin TRM if again you directly relocate to the tissue, showing again that the tissue can really dictate the function and cytokine dependence of TRM populations. So again, we used our trick of isolating liver TRM from previously sort of transferred mice that have large populations of liver TRM embedded within that tissue. We isolate them and transfer them directly in through the skin. And what we find, if we just look 10 days later at the skin of these ex-liver TRM, you can see that here they're regulating CD103, and indeed they can be found within this tissue. And more so than that, once these ex-liver TRM get into the skin, if we look at transcriptional changes, upregulation of molecules like PD-1 and E-cadherin, and suppression of transcription factors, which are specifically associated with needing to be downregulated for skin TRM sulfate, such as the OMES and TBET. And if we repeat this experiment now using wild type and knockout cells that were embedded within the liver, and remember, our liver TRMs that can't respond through TGF-beta are generally just fine, and we look within the spleen and skin now, if you're an ex-liver TRM that can or can't respond to TGF-beta, and we then re-isolate you from the spleen, these cells are absolutely fine. But if you go and you look in the skin now, if these ex-liver TRM can't respond through TGF-beta, they rapidly decline in numbers, and again, can't put on that characteristic skin phenotype, showing here a lack of CD103. So, as I mentioned, all this work has been sort of a labor of love by Susan Christo in the lab. And I'd really like to kind of leave this work with the following notions, that TRMs are really not created equal, and we can find them on a whole spectrum of behavioral states. So we know indeed that TGF-beta is a major driver of differences in TRM cell phenotype, and we find this extends beyond organs, beyond the skin and the liver, where we find that the TGF-beta responsive population exhibits a dampened functionality. And I didn't have the time to talk about this today, but also it affects their differentiation potential. And we find that TRMs exist within different maturation states, whereby those TGF-beta responsive TRMs that express CD103 have more of that handbrake on them, appear to be functionally restrained, but also have a higher longevity within the tissue. So just to wrap up this talk with some sort of broader conclusions, hopefully I've shown you today that TRMs can provide local immunity in a range of settings against both infection and cancer, and they're also associated with a wide range of skin pathologies. I've shown you that TRMs have a distinct developmental program, the shutdown of tissue egress being key for these cells, and their transcriptional identity is distinct compared to those found within the circulation. I've shown you that TRMs within different tissues have distinct requirements for their formation and survival, and the local tissue imprint and the environment on the tissue can have major influences on TRM behavior and function. And of course, it would be amiss of me not to mention that this really isn't the whole part of the story. Yes, the microenvironment is critically important, but there's another side to the story, which is work currently being done in the lab by Simone Park, where even within the same tissue, you can have a lot of heterogeneity of distinct TRM subsets. So even though there is a layer by the tissue, there's also a layer that imparts discrete TRM behaviors before you even get there. So just to thank everybody involved in the work, this is my fantastic lab. Hopefully I've highlighted everybody who's done all this work along the way. I'd like to thank our collaborators, both in Melbourne and internationally. Thanks to all the funding that makes us doing this work possible. And thanks again for the invitation to present this work. And thanks for everybody for their attention for listening to this talk.
Yeah, thank you, Laura. That was spectacular. I was absorbed for the whole talk. I almost didn't realize it got darker here. It's fascinating, your work. Thank you so much for sharing. I have made questions, but you know, the questions we'll do them by Twitter, as Carla will explains, and I cannot wait to share your talk with the global IMMUNOTALK's audience this Wednesday. Yeah, so thank you so much. We really appreciate your effort, and we hope you can answer the questions via Twitter.
Yes, thank you.
Okay, perfect. So thanks, everybody. Hopefully now you can hear me, and I'll just remind you that we will use Twitter for the questions that you might have. There's already a tweet that you can find at the Global IMMUNOTALK Twitter account that says ask questions for Dr. Laura Mackay here. And just please remember that when you reply to the tweet, it's great if you can include the hashtag Global Immuno, and Laura will use her own lab account to answer your questions. So thanks everybody for joining, and we look forward to seeing you next week. Take care. Bye-bye.
Bye.